Today, we will discuss the third section of membrane transport, which is Symport and Antiport systems. The purpose of today's lecture is to learn what secondary active transport or co-transport is, to discuss the processes of the two types of secondary active transport, which is Symport or Antiport, and how cells utilize various co-transporters to carry out important physiological functions. Let's look at the introduction first. Symport and Antiport transport systems make up the secondary active transport, which is a form of active transport across a biological membrane in which a transporter protein couples the movement of an ion, which is typically sodium or hydrogen ion, down its electrochemical gradient to the uphill movement of another molecule or ion against a concentration or electrochemical gradient. Thus, energy stored in the electrochemical gradient of an ion is used to drive the transport of another solute against the concentration or electrochemical gradient. Secondary active transport is also commonly referred to as co-transport or ion-coupled transport, wherein coupling between the transported molecule and co-transported ion is obligatory. That is to say that both the transported and co-transported species must be bound to the transporter for translocation across the membrane to occur. Unlike in primary active transport, in which ATP hydrolysis provides the free energy needed to move the solutes against the concentration gradient, in secondary active transport, the free energy needed to perform active transport is provided by the concentration gradient of the driving ion. So to call this process secondary active transport is appropriate, since the existence and maintenance of concentration gradient of the driving ion is accomplished by primary active transporters, that is pumps. So in figure 1, in Symport, the direction of transport is the same for both the driving ion and the driven molecule or ion, whereas in Antiport, the driving ion and the driven ion or molecule are transported in opposite directions. X and Y represent transporter substrates. Now let's look at Symport transport systems first. Mammalian cells express many types of sodium-coupled symporters. The human genome encodes literally hundreds of different types of transport proteins to transport a wide variety of molecules into cells against their concentration gradients. However, some symporters use an existing hydrogen ion as the driving ion instead of sodium ions. So let's study some examples of Symport transport systems. First, sodium-glucose symporters. Many cells, such as those lining the small intestine and the kidney tubules, need to concentrate glucose against a very large concentration gradient. Such cells utilize a 2-sodium, 1-glucose symporter, a protein that couples transmembrane movement of one glucose molecule to the transport of 2 sodium ions. By transporting 2 sodium ions per glucose, this sodium-glucose Symport protein can accumulate glucose against a much steeper concentration gradient than if only 1 sodium ion were transported per glucose. So this Symporter also recognizes galactose as a substrate. Its function is crucial to dietary glucose and galactose absorption in the small intestine and reabsorption of filtered glucose and galactose in the proximal tubule of kidney nephrons. Cells use similar sodium-powered symporters to transport substances other than glucose into the cell against a high concentration gradient. Several types of sodium amino acid symporters allow cells to import many amino acids into the cell. Other examples of sodium-powered symporters found in many different cells and tissues performing a variety of important physiological functions are shown in this illustration. The sodium phosphate cotransporter, also known as NAPI, is found in the apical membrane of epithelial cells of small intestine and renal proximal tubules. It utilizes the sodium electrochemical gradient to drive the uphill transport of inorganic phosphate into the cell. So for every transport cycle, NAPI couples the inward translocation of 3 sodium ions and 1 divalent phosphate. Next is the sodium iodide symporter, or known as NIS, and this is responsible for the accumulation of iodide from blood by the thyroid gland, which is absolutely necessary for the synthesis of thyroid hormones. Here, it is localized to the basolateral membrane of thyroid follicular cells and co-translocates 2 sodium ions and 1 iodide ion per transport cycle. Here, NIS is also found in other tissues, such as in the mammary glands, where it functions to transport iodide into the nursing mother's milk. Then we have the sodium-potassium chloride cotransporter, the sodium chloride cotransporter, as well as the potassium chloride cotransporter. And they belong to the same family of transporters and are all electroneutral cotransporters. The sodium-potassium chloride cotransporter, or NKCC, utilizes the sodium electrochemical gradient to drive the inward co-translocation of 1 sodium, 1 potassium and 2 chloride ions. Among other places, NKCC is found in the apical membrane of the thick ascending limb of the loop of Henle, where it performs an important role in urine concentration. The sodium chloride cotransporter, NCC, utilizes the sodium electrochemical gradient to drive the chloride transport into the cell with 1 sodium is to 1 chloride coupling stoichiometry. NCC is found in the apical membrane of the epithelial cells of kidney nephron distal tubules. Likewise, the potassium chloride cotransporter, KCC, translocates 1 potassium and 1 chloride ion out of the cell per transport cycle. Here, the driving ion is potassium ions. Additional examples of cotransport proteins include the GABA transporter, also known as GAT, GAT, which couples the inward translocation of both sodium ion and chloride ion to the simultaneous inward translocation of GABA. It regulates the basal concentration of GABA in the nervous system and, in addition, regulates the concentration and lifetime of GABA in the synaptic cleft. The hydrogen ion monocarboxylate cotransporter, MCT, a proton-driven transporter which recognizes monocarboxylates such as lactate, pyruvate, acetoacetate, and beta-hydroxybutyrate. The hydrogen ion oligopeptide transporter, PEPT, it couples the downhill movement of 1 hydrogen ion to the uphill movement of 1 dipeptide or 1 tripeptide and is located in the apical membrane of the small intestine, epithelial cells, and renal proximal tubules. PEPT is thought to be the dominant player in nitrogen absorption in the small intestine and nitrogen reabsorption in the kidney tubules. Then there are 2 sodium-coupled bicarbonate transporters, NBC, which perform important acid-base balance functions. The direction of transport may be inward or outward. For example, NBCE1, located in the basolateral membrane of kidney proximal tubules, mediates the outward translocation of 1 sodium ion and 3 bicarbonate ions into the interstitial fluid, where the reabsorbed bicarbonate is returned to the circulation by entering the peritubular capillaries. Thus, NBCE1 plays a crucial role in renal bicarbonate reabsorption. Now let's study some examples of antiporters, which are also known as exchangers. The plasma membrane of most cells contain one or more types of antiporters, which couple the movement of a co-transported ion, which is often sodium ion, down its electrochemical gradient to the movement of a different molecule in the opposite direction against a concentration gradient. In cardiac muscle cells, for example, a sodium-calcium ion antiporter rather than a plasma membrane calcium ATPase plays a principal role in maintaining a low concentration of calcium ions in the cytosol. Now note that the movement of 3 sodium ions is required to power the export of 1 calcium ion against a very high electrochemical gradient between the cell interior and the cell exterior. The operation of the sodium-calcium antiporter, it lowers the cytosolic concentration of calcium ions and reduces the strength of heart muscle contraction. The sodium-potassium ATPase in the plasma membrane of cardiac cells, as in other body cells, creates a sodium concentration gradient used to power the export of calcium ions. The drugs wabane and digoxin increase the force of heart muscle contraction and are widely used in the treatment of congestive heart failure. The primary effect of these drugs is to inhibit the sodium-potassium ATPase, thereby raising the intracellular sodium concentration. Because the sodium-calcium antiporter functions less efficiently with a lower sodium ion concentration gradient, fewer calcium ions are exported, and so the intracellular calcium ion concentration increases. This increase causes the muscle to contract more strongly. Now let's look at the sodium proton exchanger, or the NHE, or you can call it the sodium hydrogen ion exchanger. Here again, the sodium electrochemical gradient is utilized by the antiporter to extrude protons from the cell, thus raising the cytosolic pH. The cytosolic pH is also regulated by another anion transporter. We call it the chloride bicarbonate exchanger, and this exchanger catalyzes the 1-for-1 exchange of chloride and bicarbonate across the plasma membrane. The direction of transport by the chloride bicarbonate exchanger is governed by the concentration gradients of chloride and bicarbonate. For example, in red blood cells, erythrocytes, traveling in the systemic capillaries, excess production of bicarbonate in the cytoplasm, which is as a result of carbon dioxide hydration reaction catalyzed by carbonic anhydrase, it drives the outward translocation of bicarbonate in exchange for inward translocation of chloride ions. When the red cells pass through the lung capillaries, the direction of transport is reversed such that inward translocation of bicarbonate is coupled to the outward translocation of chloride ions. The bicarbonate that enters the cells ultimately gives rise to carbon dioxide, which is catalyzed by carbonic anhydrase, which then leaves the cell and ultimately the capillaries to enter the air-filled alveoli of the lung, where it is then exhaled into the atmosphere. Numerous transport proteins also enable plant vacuoles to accumulate metabolites and ions. The lumen of plant vacuoles is much more acidic, which has a pH of 3 to 6, than in the cytosol, which has a pH of 7.5. The vacuolar membrane contains a V-class ATP-powered pump and a unique PPI-powered pump, both of which function to pump protons into the vacuolar lumen against a concentration gradient. So, as illustrated in this figure, the vacuolar membrane also contains chloride ions and nitrate channels that transport these anions from the cytosol into the vacuole. Entry of these anions against their concentration gradients is driven by the inside positive potential generated by the hydrogen pumps or the proton pumps. The vacuolar membrane contains two types of proton pumps, which is a V-class proton ATPase, which is given in light green, and a unique pyrophosphate hydrolyzing proton pump, which is given in dark green. These pumps generate a lowered luminal pH as well as an inside positive electric potential across the vacuolar membrane owing to the inward pumping of hydrogen ions. The inside positive potential powers the movement of chloride and nitrate ions from the cytosol through the separate channel proteins, which are given in dark purple here. Proton antiporters, which are shown in light purple here, powered by the hydrogen gradient, accumulate sodium ions, calcium ions, and sucrose inside a vacuole. The proton gradient and electric potential across a plant vacuum membrane are used in much the same way as a sodium gradient and electric potential across the animal cell plasma membrane. That is, to power the selective uptake or extrusion of ions and small molecules. In the leaf, for example, excess sucrose generated during photosynthesis in the day is stored in the vacuole. During the night, the stored sucrose moves into the cytoplasm and is metabolized into carbon dioxide and water with concomitant generation of ATP from ADP and inorganic phosphate. A proton sucrose antiporter in the vacuolar membrane operates to accumulate sucrose to plant vacuoles. The inward movement of sucrose is powered by the outward movement of protons, which is favored by its concentration gradient, and by the outward negative potential across the vacuolar membrane. So, uptake of calcium ions and sodium ions into the vacuole from the cytosol against their concentration gradients is similarly mediated by proton antiporters. So that was all about Symport and Antiport systems. Before we conclude, let's go through some of the major points that we have come across so far. Symport and Antiport transport proteins belong to a discrete class of proteins called cotransporters that use the energy stored in the electrochemical gradient of sodium or hydrogen ions to power the uphill movement of another substance in a process called secondary active transport or simply cotransport. Entry of glucose and amino acids into certain cells against their concentration gradient is via sodium-coupled symporters. Two proton exporting transporters, a sodium chloride bicarbonate cotransporter and a sodium proton antiporter, maintain the cytosolic pH in animal cells very close to 7.4 despite the metabolic production of carbonic and lactic acids. A sodium-independent chloride bicarbonate antiporter, similar to AE1 protein, functions to export bicarbonate when the cytosolic pH rises above normal, causing a decrease in pH. In cardiomuscle cells, the export of calcium ions is coupled to the import of sodium ions by a cation antiporter, which transports 3 sodium ions inward for each calcium ion exported. Then the erythrocyte membrane contains a chloride bicarbonate anion exchanger or antiporter, which is commonly known as the AE1 protein that facilitates transport of carbon dioxide by the blood. Uptake of sucrose, sodium ions, calcium ions, and other substances into plant vacuoles is carried out by proton antiporters in the vacuolar membrane. Ion channels in the membrane are critical in generating a proton concentration gradient large enough to power accumulation of ions and metabolites in vacuoles by these proton antiporters. So that was all about Symport and Antiport systems. We hope this lecture was helpful in understanding the concept of these transport systems and their importance to living cells and organisms on the whole.